Okay, let's get started. Good day, everyone, and thank you for joining us. The webinar today is entitled CITE-Seq Identifies Novel Stromal Cell Populations in Diseased versus Healthy Human Tendon. My name is Jeff Jensen, and I will be acting as your host for today's event. First things first, we want to thank BioLegend for sponsoring this webinar. You know, companies don't have to do things like this. They could just run a print ad or a commercial somewhere, but BioLegend takes the extra effort to bring content like this to researchers around the world. And the fact that so many of you have registered and are here watching today means that this is obviously a valuable thing for the research community. So we can't thank BioLegend enough for making these webinars happen. And if you appreciate content like this, I would encourage you to head over to BioLegend's website to check out the massive content library that they have put together. It is super useful. The webinar today is being hosted by FluoroFinder. If you frequently use multi-parameter flow cytometry in your research, then you should get yourself familiar with FluoroFinder, the gold standard of online panel design tools. More information can be found at fluorofinder.com. As a reminder, today's webinar is a live event. There will be a question and answer period at the end, but please feel free to ask questions during the presentation by using the GoToWebinar control panel located on the right side of your screen. The webinar is being recorded, and a link to the recording will be sent out to all registrants. Our presenter today will be Dr. Adrian Kendal. Dr. Kendal is a specialist surgeon in trauma and orthopedics with subspecialty in foot and ankle reconstructive surgery, and a lecturer in organization of the body at Trinity College, Oxford. His research aim is to understand the pathogenesis of chronic debilitating tendon disease. His team applies combined single-cell transcriptomics and surface proteomics to healthy and diseased human tendons, identifying novel tendon cell subtypes. For the first time, they have shown that human tendon harbors at least 5 distinct COL1A1-2 expressing tenocyte populations, in addition to endothelial cells, T cells, and monocytes. And now, Dr. Kendal, if you are ready, I'll go ahead and pass control of the screen over to you so you can start today's presentation.
Thank you very much. I'm delighted to be a part of this. It's a really exciting thing, and I've been greatly supported by BioLegend in this work. So, by way of a brief introduction, tendon degeneration and tendon disease is an increasing problem globally, particularly as we start to see an aging population across the world, but more importantly, a population that wants to stay active for longer. Tendon disease can affect any part of the body, but it typically tends to affect the foot and ankle. In fact, that's the most common site. At least 20% of the population at any one time will suffer chronic tendon pain. That's different to a tendon rupture or acute tendonitis, which normally resolves. And that's an incidence of about 18 per 1,000 person years. Again, the Achilles is most commonly affected. It's also the most commonly ruptured. And it's worth saying that as well as the pain and disability caused by tendinopathy, because these are tendons that cross the small weight-bearing joints of your foot and ankle, it is often a precursor to joint deformity, degeneration, and ongoing destruction. And this is an area that we have yet to identify any solid curative treatments. Our hypothesis is that in the context of chronic tendinopathy, there are distinct pathogenic cell populations that influence the normal behavior of healthy tendon cells. And this may occur at key sites, for example, perivascular or interfascicular locations. Just to set this recent study up in previous work, we struggle with these two questions. That firstly, how do we how do we describe normal tendon cell behavior, particularly in vitro? And when we're studying these cells in vitro, are we looking at one single tendon fibroblast, or are we looking at multiple different subtypes? And it's worth pointing out that up until recently, most people would identify and define a tendon fibroblast as any cell that's explanted from tendon which can survive on a culture surface and adhere to that surface, and any cell that has a spindle shape. And if you compare that to, for example, immunological definitions of different types of T and B subsets, you can see there's a lot of work to be done in trying to refine this area. In terms of in vitro assays, it's important to understand that tendon fibroblasts are exquisitely sensitive to their surrounding environment, and in particular to the surface that they contact. And more we're seeing that if we want to study these cells in vitro, we need to be looking at them on scaffolds that at least mimic normal tendon architecture. There's lots of different ways of doing this, but the Carr Group in Oxford has focused on using electrospun fibers and aligning those fibers to replicate normal tendon shape. You can use any fiber, but we've used polydioxanone, which is PDO, and that's because it's got a very good safety record as a suture material in patients. It can also be produced as a polymer solution, and that means that across a high potential difference, you can begin to spin very thin and narrow fibers. And the other thing is it's water dissolved, it's water soluble, it's absorbed over time in the human body, so it's a useful material if you're designing, for example, a temporary patch to help tendons heal. The electrospinning process is really impressive, and if you have a look at that scanning electron micrograph on the top right, going diagonally across the image is a hair, and either side of that are these very fine spaghetti fibers, which are the PDO electrospun fibers. And what we found quite early on is if we put tendon cells, either from diseased or healthy human tendon, onto these electrospun fibers, particularly when they're arranged in series, the cells respond to that mechanical environment. So here are two tendon cells that have been dyed with a green fluorescent protein, and running almost vertically up and down the image are these very fine PDO fibers. But you can see that within hours, they respond to that surface, and they use the fibers as a scaffold, as a network to spread out along, and they then migrate along these fibers. And it's a really impressive change in morphology. I mean, they start off looking like poached eggs, and after a few hours, they can be anything up to about 20 micrometers in diameter and 200 plus micrometers in length. So what we found from this early work was that actually even disease cells were able to respond to these aligned PDO fibers, and that gave some credence and hope to some implantable meshes and scaffolds that we could use in patients who've got chronic tendon rupture. They didn't respond in the same way, the same dynamic profile as healthy tendon, and although they proliferated, it was at a reduced rate, and they produced less of the reparative collagen than healthy cells. But one of the difficulties with this work is that firstly, we were comparing different tendons from different patient groups. So the healthy tendon cells were explanted from a hamstring tendon that was excess, was left over from a hamstring graft used to reconstruct a knee ligament. So typically an anterior cruciate knee ligament. This is a common operation, and it tends to be young patients, often young male sportsmen in their late teens, early 20s. And they're a very different group from the patients where we were able to obtain excess or wasted tendon tissue from disease rotated cuff. That's a tendon of the shoulder. And again, that patient group was tended to be women predominantly over the age of 45, up to the age of 70. Secondly, I think there's quite a bit of heterogeneity within tendon cells, and that subsequent work showed that, but I was never confident that we were looking at the same cell population from one experiment to the next. So some cells would adhere, for example, to the fibers and some wouldn't. Is that because they're actually just different cell types? And even if they're all the same cell type, we know that there's a lot of difference in their shape depending on the surface environment. And this makes traditional methods such as flow cytometry quite difficult. You have quite a wide range of forward and side scatter. Fibroblasts tend to autofluoresce, particularly tendon fibroblasts. And all of this adds up to a difficulty and an inability to identify and isolate cells of interest in vitro and ex vivo. On top of that, tendon is a very difficult tissue to work with. My early work was using regulatory cells and T cells. And actually, immunology is much easier. You have plenty of cells normally in a sample, and they're used to being in a cell suspension. Tendon is very different. There are very few cells, particularly in a healthy tendon. You can see that from this histology image on the top left. And what few cells there are tend to be completely engulfed by a very dense collagen matrix. in addition, they're tightly adhered to that matrix. It's difficult to prise them away when you're trying to explant them and work with them in vitro. We also don't have a good model for chronic tendon disease in animals. We've got a lot of good models for acute disease and also rupture, but not for chronic disease. And as I said, they're so sensitive to their surface environment that I worry that a lot of what we observe in vitro is misleading and doesn't necessarily represent what's happening in normal human biology. So the overall aim of the work, therefore, based on this early work, was to answer these three questions. So firstly, I'll be looking at one particular type of tendon fibroblast, or are there multiple subtypes? Is there a difference in the composition of tendon cell subtypes in disease versus healthy human tendon? And how might these differences be regulated? And based on all the limitations I've spoken of up until now, this is where CITE sequencing is so useful. So not only do you get the single cell RNA sequencing and transcriptomics information, but you can get a readout of surface proteomics. And we perform this in conjunction with the Wellcome Trust Centre for Human Genomics here in Oxford. And I'm sure people have seen this before, but I think this is quite a useful cartoon. And what is so good about CITE sequencing is the surface proteomics readout. So it's a great idea. This is the kind of thing I wish I'd thought of, where any antibody that binds to a surface protein or a target that has a good pedigree and good track record can be conjugated to an oligonucleotide barcode. And this then means that you can then run a sample in the same way that you normally would, for example, using a 10x platform. And the poly-A tail of that oligonucleotide barcode can match up with a poly-T associated with the, for example, 10x gem. And in the same way that you'll get a readout for the single cell transcriptomics, you can get a readout for that surface proteomics. And in a really simple way, this is the way I see CITE sequencing. It allows you to do three really useful things. As I said, you get your single cell RNA sequencing data. You can take any antibody that's conjugated in this way that recognizes the surface protein. So for example, if you're looking at immune cells, you can use anti-CD4, anti-CD8, etc. And then thirdly, you could take antibodies that recognize ubiquitous surface proteins, for example, beta-2 microfibril or ATPase. And so long as they are conjugated with one of 8 different barcodes, you can then run effectively cells from up to 8 different tendon donors or sample donors in a single lane of sequencing. And this has 2 huge advantages. Firstly, it massively reduces the cost. So for example, 24 samples can be run for almost as much as 1 lane of sequencing. And secondly, you can greatly reduce your batch effects by having a single sequencing event. When we started this work, it wasn't clear whether it would be applicable to tendon, again, for all the difficulties with working with this tissue. So we flung the net really far and wide, and this was the first time we were able to get CITE sequencing to work in humans. In terms of the healthy human tendon tissue, we had a number of different donors. Most of them were having tendon transfer surgery, either across the foot and ankle, where a healthy tendon was moved from one side of the foot to the other side to try and balance often a neurological problem or a neurological imbalance. And that included tibialis posterior tendon and the flexor hallucis tendon that goes to your big toe. And again, we had a large number of hamstring donors, and these were the typically young sports people who were undergoing anterior cruciate ligament reconstruction. All of this tissue was obtained using our under our local ethics, and all of this was tendinous tissue that would normally be disposed of, it's normally surplus to requirement. The diseased human tendon samples came from 3 main sources, but in all cases, it was in patients who'd had chronic tendon pain and dysfunction lasting at least 12 months. And in all cases, the pain was not helped by any non-operative measures, whether that's physical therapy, a change in footwear, change in lifestyle, analgesics, anti-inflammatories, injections. No matter what they tried, they still had debilitating pain. And as a result, they were considering surgical exploration, debridement, and in some cases, excision of the painful tendon. So our samples included 3 Achilles tendon samples, 2 peroneal tendinopathic tendons, and 6 toe extensor tendons that run over and associated with this deformity of this joint here, the proximal interphalangeal joint. And you often see that with what's called a hammer toe deformity of the toe. In terms of preparing the samples, again, these are tissues with a lot of matrix and little cell composition, so it takes longer to manage to actually digest the extracellular matrix sufficiently to get the cells. But we managed to optimize a process where after about 90 minutes from the minute we received the sample from patients, we had a cell suspension, and that used a combination of mechanical and enzymatic digestion. All the samples were then cryopreserved at -80 degrees. They could then be further processed before sequencing and then run as a batch. And again, because they're all hashed with up to 8 of the different hashing antibodies, we were able to run 8 samples per lane of sequencing. In terms of preparing the samples for sequencing with reference to the CITE sequencing, this has been well described, and there's a reference there. And we essentially followed this recipe, but it's, to be honest, it's no different to preparing cells for flow cytometry. So the cell suspension is blocked, is incubated in FC block buffer, and then washed, and then the surface antibodies are added for incubation. That's washed again, and then ultimately each sample from each particular donor is hashed with one of the 8 hashing antibodies before the whole lot are mixed together. You might say, well, what were your surface targets? And these were the dozen or so monoclonal antibodies that we used to recognize surface proteins. And part of the reason for using these was because these are the ones that were available from BioLegend, and I know that repertoire is increasing all the time. But we wanted a collection of markers that had some evidence in the literature for identifying MSCs and some muscle cells. We wanted a collection that could identify fibroblasts, not necessarily musculoskeletal fibroblasts, but fibroblasts in general. So we had CD73, 10, 26, and 44. We wanted to be able to identify and in some ways exclude from our analysis hematopoietic and lymphoid cells. So we used CD95, 54, 55, 45, and CD9. And we had 2 surface markers at the bottom there just to identify endothelial cells. So that's CD31 and CD34. In terms of the analysis, I used the CIRAP package on with the R platform. And the key here was being able to select on samples based on the hashing barcode readout, and then becoming confident that you had cells that had all come from a particular tendon donor, and then integrating those samples together before a final transform and then the PCA dimensionality reduction. And this is the kind of raw data that, or early processing data that we received. So you can see each column represents the cells from a particular sample. Again, the numbers here are small compared to a lot of other single cell sequencing work, but that just reflects the paucity of cells in tendon tissue. And you can see that those cells that have come from the sample hashed with antibody 1 and 2 are very few numbers, so they were excluded from any further analysis. We had decent numbers from samples 3, 4, 5, 6, 7, and 8. What's nice about this again is it's very easy to pick up doublets, which is an issue with some of the single cell work. Similarly, we excluded any cell that had evidence of multiple hashing antibodies binding on the surface, and any cells that didn't have any antibody binding on the surface. With that raw data, we could then have a look in more detail and actually refine our strategy for isolating cells from a particular donor. So, for example, here's a scatter graph showing on the y-axis any cell that has bound to a hashing antibody 4, and on the x-axis hash antibody 1. And if we select that group based on those parameters outlined in the hashed blue square, and then have a look, you can see that all those cells have very little surface antibody, hashing antibody 1, 2, 3, 5, 6, 7, and 8, and they have a lot of hashing antibody 4. So this is an example of how we can be confident that those cells will come from a particular donor. And if there's any doubt, then the cell was excluded from analysis. Once those hash samples were all integrated, the data from those hash samples were all integrated, we were able to look for clusters based on unbiased transcriptomic differential gene expression analysis. And you can see that in diseased and healthy tendons, there are a number of different groups. These included monocytes, T cells, and endothelial cells. But there were 5 clusters that all expressed high levels of collagen type 1 genes and also collagen type 3. And these were labeled tenocyte A, B, C, D, and E to begin with. As everyone's aware, we can look in more detail at the differential gene expression of those particular tenocyte clusters compared to each other, and we begin to see patterns such that tenocyte A has increased expression of PENK, KRT19, PTX3, SERPINE2. Tenocyte B has high levels of periostin and KRT7. Tenocyte C looked like a group of recently described smooth muscle mesenchymal cells in the murine data set that express high levels of ITGA7, ACTA2, and MYL9, and they're associated with pericytes in the perivascular region. So it's interesting to find them in humans. D looked like a bunch of fibroadipogenic tenocytes, and tenocyte E had similar gene expression profile to these TPP3 positive, PGR4 positive cells, again identified in a mouse data set of patellar tendon that demonstrated some pluripotent characteristics. So, having started with very little information as to different subtypes, we were beginning to be able to identify at least 3 or 4 subtypes based on this differential gene expression. One of the concerns always with single cell work is that what we're looking at, the clusters we're looking at really are not the result of any biologically relevant differences, but are the result of some confounding transcriptomic variation. So we decided to have a look at our data set, but coming from the other extreme and saying, well, what are all the proteins that are normally found in tendon? And if we have a look at the genes that code for those proteins, how are they distributed amongst these 5 groups? So fortunately, Hakimi, Osman Hakimi and the group have previously catalogued the common proteins found in healthy and diseased tendon. And when we took the top 50 genes expressing those proteins and matched them onto our data set, we found that firstly, no single tenocyte cluster expressed all of them. And again, there was quite a clear demarcation of those 50 genes across the 5 groups. So again, this suggests that there may be some biological relevance to these different clusters. And it also allowed us to start to identify them in more detail. So again, that tenocyte C group has high expression of TPN2, Taglin, MYL9, ACTA2, and as I said before, ITGA7, all of which fit with the murine data set. Similarly, we can see a group that has high expression of the corin, LUM, fibrinogen, and COMP, and PREL, which we think are very similar to this TPP3 positive cells. So I think the stromal world will at some point settle down, and we'll be able to, with more information, identify these and describe these in more detail. But at the moment, we labeled these clusters in this way. So we think tenocyte A are a group of PTX3 positive or high expressing cells. Tenocyte B express high levels of Scraxis and KRT7. Tenocyte C are ITGA7 positive, and D are APOD positive, and E, as I said, are these TPP3 positive cells. Well, what was pleasing was that when we stained for the protein products of some of these genes in healthy and diseased human tendon tissue, we were able to see examples of these cells on histology. And again, this lends some weight to the idea that there may be genuine biologically relevant cell subtypes. As I said at the beginning, a huge advantage of CITE sequencing is a surface proteomic readout, and one of the keys here is to try to be able to identify and isolate live cells based on surface markers. And even with quite a limited repertoire, we're beginning to see some useful patterns. So for example, if we look at the tenocyte A group, they express high levels, so darker black dots, of CD10, CD26, and CD54. And this is the protein, not the gene, that's being that's binding to the monoclonal antibody with the oligonucleotide barcode conjugators. This is the protein on the surface. You can see at the bottom two images on the towards the right-hand corner, the endothelial cells, as expected, have high levels of surface CD34 and 31. These tenocyte C, SMMC type cells have got high expression of CD146 directly in the middle of that 3x3 grid. And so we're beginning to build up a bit of a picture where we can perhaps start to identify live cells based on surface markers. And we're just going to focus here now on the tenocyte A group. You can see with this co-blended plot with antibody recognizing surface protein CD26 and surface CD54. The white dots are cells that express both. And again, there's a large proportion that seem to express both those surface proteins. Similarly, focusing in on these PTX3 positive cells, the majority seem to come from diseased tendon. And interestingly, we know that PTX3 in endothelial cells, other fibroblasts, and immune cells has got some evidence of increased expression in response to pro-inflammatory stimuli and also LPS. There's only one previous description in a rat model of tendon, but here we see high expression of PTX3, a large proportion of those cells in diseased tendon. And interestingly, all the cells that are PTX3 positive from disease samples seem to upregulate the chemokine genes CXCL1 and 6, and also cytokine IL-8, so CXCL8. When we look for co-expression, we see that there's a very small group that co-express all three of those pro-inflammatory genes. And we think these might be some of the cells that are driving that chronic tendinopathy process. We can do a similar analysis for the other groups, and you can see that, for example, tenocyte B from diseased tendon has increased regulation of some genes such as KRT14, 19, and COMP. Tenocyte C seems to express more reparative collagen type 1 and 3, and also basement membrane 4 and Thi1 in diseased tendon. D seems to upregulate Thi1, CXCL2, ICAM1, and also reparative collagen type 3, again, as you'd expect in a disease situation. And even the endothelial cells that have come from diseased tendon have increased expression of selectin E, ICAM1, CCL2, CXCL12. So as you might imagine, in this pro-inflammatory, chronically inflamed and painful tendon, we see an increase in some of these inflammatory signatures. We focused on the tendon cells, but clearly there are immune cells within healthy and diseased tendon. In the diseased tendon, it seems to be that macrophages dominate. There are a few dendritic cells. And again, as you'd expect, when we look at the macrophages from diseased tendon versus healthy tendon, we see an increase in chemokines, CXCL3, cytokine CXCL8, CCL2, and so on. And in dendritic cells, we see an increased expression in lysozyme gene, some of the complement genes, C1QA and QC. So in conclusion, we can now say that CITE-Seq and also cell hashing does work for human tendon cells. It's a challenging tissue to work on, but we've been able to apply it. We've shown for the first time that actually both healthy and diseased tendon are indeed made up of a number of different tendon cell subsets. Future work will be based on seeing if we can isolate these and explore their stability or their plasticity, particularly when we look at them in vitro. As we expected, we see an increase in the inflammatory signal, particularly in diseased tendon, across all those tenocytes, not just the innate immune cells and not just the endothelium, but also the tenocytes. And in particular, we think that the PTX3 positive cell subset, which seems to predominantly express CXCL1, 6, and 8 in the disease setting compared to the healthy setting, might be a useful avenue for potential therapeutics in the future. We're still trying to identify robust surface markers by which we can isolate and interrogate some of these cell types in the future. And again, I think as we see an increase in the repertoire of those surface oligonucleotide barcoded antibodies, we should be able to fill in some of those gaps. Single cell sequencing work is very vulnerable to confounding variation, particularly between individuals and particularly between different anatomical sites. And ideally, what we really want to be able to do is identify disease versus healthy differential expression within the same individual. And I think there are ways in which we can sample normal and diseased tendon from the same patient, and I'm happy to talk about that. I think we're still at the stage where we're trying to identify these different cell types and also look for key signaling pathways involved in tendinopathy. But I think this is really useful technology, and it's been well used now in other areas. And I think one area in stromal biology that would be useful to apply this to is charting the kind of progress of bone marrow derived cells, particularly in vitro, as they're cultured and differentiated into tendon cells or muscle cells, and seeing which transcription factors are increased and which genes are expressed or downregulated throughout that process will be very useful, particularly when it comes to reparative medicine. So thank you very much for the opportunity to talk here, and these are all the people that helped with this work. Professor Carr was my supervisor throughout it, and I'm very grateful to my surgical colleagues who helped provide the tissue, and also the Oxford Genomics Centre who provided the sequencing. So thank you very much.
Wow, Dr. Kendal, thank you so much. That what a clear and concise explanation of some very complicated things. It's fantastic. Fantastic. I know we have a lot of questions coming in, and so let's get right to them.
Right, thank you very much for the questions here. I don't know if Jeff's happy for me just to go through some of these.
I'll go ahead and read some off to you, unless, yeah, I'll go ahead and read some off to you. And then the first one was from Ludovine. Were your surface proteins signal well correlated with the transcriptomic data for the mRNA encoding these proteins? That was the first question coming in. Okay, yeah.
Yes, they were, but of course, you can have a gene transcribed, and you just need a protein on the surface that has a longer half-life, and you'll soon see a difference in the surface proteomics, and that's obviously a problem in general with cell biology. But most of the time, we could see a differential gene expression that matched well with the differential surface proteomics.
Great. And then we had another question actually from this same person, was asking, was the ADT data crucial for the identification of your different tenocyte populations?
No, not at this stage. I mean, I think that's where we really want to be. And as I said, in many ways, it's exciting to be a part of stromal biology with the tools that we now have, because I guess it's a bit like immunology sort of 30, 40 years ago, but where we really don't have a collection of surface markers by which we can reliably identify the different cell populations, and that's definitely what we're aiming to do. And I think part of the inability to do that on this first attempt was that I don't think we had enough surface antibodies that recognize surface proteins to capture all those different cell subtypes. I think we need a couple of things. One, we need to know which cell types we're looking at, by whichever way, whether it's differential gene expression or surface proteomics. And secondly, we need a big enough repertoire of surface proteins to start, or targets to start looking at those. So if you were doing CITE-Seq on a PBMC sample, you automatically know which antibodies to go for in order to identify your subtype. But we're just not there yet, and I think I think that's really the next stage of this.
Okay, great. Thank you. We had a question coming from Lauren DeVore, and I apologize if I'm pronouncing your name incorrectly, Lauren. She said, thank you very much for the great talk, incredibly interesting. Was it difficult to establish the mechanical and enzymatic digestion for sequencing?
Yeah, it really was, and I think again this is a big limiting factor when it comes to stromal research, because as I sort of laboured already, there aren't many cells, and they're so tightly adherent to these stromal structures and extracellular matrix that it is difficult. It's a process that we're still trying to optimize, if I'm honest, and it was very much hit and miss, because you have to use proteases. We use liberase because it's slightly less toxic to the cells than other forms of collagenase. We use some DNAs as well to help, but more than about 45 minutes you started to see the cells die. Less than that time, they didn't seem to come away from the matrix sufficiently. There's clearly a role for how well you mechanically chop up the tissue before you add the proteases. And actually, one thing you can do is have a look under a microscope at your samples throughout that sort of 45-minute to 1 hour period. You can begin to see some of the cells come off, and if you leave it too long, you can start to see them. You can start to see that you're losing them. So it is difficult, and I'd also say that I tried mouse tendon samples to optimize it, but actually it's an awful lot easier. I found it an awful lot easier to get good cell yield from murine tendon than I did from human tendon. So particularly healthy tendon is very difficult. So it is hard, and that's why you see such smaller numbers compared to other areas of biology.
Fantastic. And we have lots of time till the top of the hour here, and the questions are really rolling in. So Dr. Kendal, if you're okay, I can just keep on reading through these. And
yeah, of course.
Great. We had one coming from Renee Svensson asking, do you have any idea which of the cell populations will dominate in culture?
Yeah, it's a really good question, and we're going to bring out some work on that shortly. One of the things we're interested in is looking at CD90 positive surface CD90 positive cell populations compared to those that are negative, and we see quite a lot of those dominate in cell culture, but we knew that before this ex vivo work. Again, you have to define what you mean by culture, because I think that growing these tendon cells on culture plastic is probably unfair. It really stretches the cells out. It's a huge mechanical queue with massive micro forces across the cell, and it's probably not a similar environment to their normal environment. And when we grow these cells on fibers, one thing we see is that those sort of pericytes, the ITGA7 SMMCs, however you want to call them, we seem to lose those, and we seem to get more of the cells that express high levels of COMP and high levels of collagen 1 and 3. So more of the sort of the TPP3 PRG4 positive type cells. But I would imagine a situation where if you change that culture environment, you start to see different cells coming through.
Fantastic, excellent, thank you. We have a few questions coming in from Megan Morgan asking if cell hashing is different from indexing inside of GEMs, and also asking about antibody tagging. And I don't know. There was no.
So so hashing, I mean, I appreciate that the matrix is a bit difficult. Hashing for me is simply using an antibody that recognizes a ubiquitous surface protein, and attached to that you can have up to however many barcodes you want, but we used 8. And that's just a way of identifying all the cells from a particular sample and running the whole lot as a single lane. So you can run up to 8 samples in one lane, for example. What's nice about the 10x gem system is that it allows for this, or it can be kind of used in this way, such that you'll get an HTO or a hashing readout as well as your surface proteomics, as well as your single cell RNA sequencing. So I don't think it's quite the same. I think it utilizes the gem system. I'm not sure if that was a question. And what was the last part? I didn't quite understand that.
It's about antibody tagging. I think you've covered it, but I'm not sure. So Megan,
yeah,
if you're out. Oh, go ahead.
I'm happy, Megan, if that has an answer, I'm happy to come back to it. I think it's just as simple as saying, well, rather than having one antibody with one oligo that recognizes a surface target, let's have 8 different antibodies with different oligos that recognizes the same ubiquitous surface target, and let's use that just to label all the cells from a particular sample.
Okay, great. And then we had a question from Nazgul Haddadi sorry if I pronounced that again. I was wondering if there would be any difference in results from fresh samples versus cryopreserved samples, and how long we can keep the samples in cryopreserved fluid.
Yeah, so it's a really good question. I can't tell you exactly how long we can keep them in cryopreservation. I don't think the length of time matters too much, but you're right, and we do lose cells as we freeze and then thaw them out. We know that, and I'll say that we probably lose about 30 up to 30% of the cell population. And as we mentioned in the publication, there is a concern that we may differentially lose some cells compared to others. So, for example, certainly some of the immune cells, some of the macrophages, tend not to survive the freezing process as well as some of the stromal fibroblast populations. If you're interested in tendon in tenocytes, that's less of an issue. But if you're interested more in the immunological side of tendon inflammatory behavior, then that is an issue. On that, I don't think CITE-Seq is a fair way of quantifying the relevant numbers and relative populations of these cells, because we simply don't know, which cells are more resistant or more hardy to processing cryopreservation and then thawing out, and which aren't. I suspect that if we were able to run these immediately ex vivo, we'd probably see a similar populations, but we may see a difference in the sort of relative percentages of each of those cells that make up each of those different groups. And it is an issue. I personally think it's an acceptable price to pay for being able to run multiple samples at once, because one of the big issues with single cell sequencing and CITE-Seq, of course, is that there's a lot of batch variation, and this is one way of trying to overcome that. But it's a really good point. So yes, we do lose cells, probably about 30%, and I think it's the thawing process, not how long they stay frozen for.
Okay, excellent. And it's sort of a follow-up question from the same person. Could you explain a little bit about the sample digestion? They're hoping to do the same thing on skin, and they're not sure if the digesting skin tissue would affect the final results. If you have any insight on that.
Yeah, so again, I think this is a difficult area, and it's one of the big stumbling blocks. But once you get over it, I think you're in a good position. So skin you may have more success, because again, it's more cellular than healthy tendon. My experience, hopefully it's helpful, is that the more you chop it up, the more you mechanically chop it up. And I'd literally use a scalpel and try and cut it into sort of 1 millimeter cube pieces at their largest. The more you do that at the beginning, the more yield you seem to get. I think at some point you're going to have to use some kind of collagenase or protease to try and release the cells. And there's definitely a sweet spot where any longer you start to lose cells. But up until that point, you need that environment to release them. Certainly for me, it was a case of trial and error for several months before we managed to get a protocol that was half workable. But it's certainly possible. And I think the mechanical side of it is really important. And I think doing it at low speed, so doing it self manually, is probably better. When we used any kind of blending machinery in the lab, we lost too many cells. I think it's too aggressive.
Excellent, thank you. And we still have many people online, and there are a lot more questions here. So if you've asked a question, we'll try to get to it here in the next few minutes, and anything we don't get to, we'll answer in writing after the webinar. But be patient, we'll get to you if we can. Thank you for answering so many questions. Appreciate it, Dr. Kendal. This one came in from Daniel Terrell. Do the antibodies have to be only target surface proteins, or can they be intracellular?
It's a good question. No, I think I don't see why they couldn't be intracellular. I think that's fine. I think you'd have to. You would have to arrange your droplet run such that you could then lyse the cells before adding those antibodies. And I think that could be difficult. And I must admit, I was not involved in the actual sequencing or the running of the sequencing. So I think there's some issues with the biochemistry of using intracellular antibodies, but I guess in theory it's possible. But I would say there are other ways of looking at intracellular proteins on a single cell basis. I mean, for example, CITE-Seq, that might be a more sensible way to go.
Okay. Great, and a good question here from Amro Hussein. Thank you, Dr. Kendal, for the excellent presentation. I wondered if you've been able to fact sort the different tenocyte populations based on surface markers, and if so, have you observed any functional differences in vitro, like in electrospun fibers, for example?
Yes, that's a really good point, and that's the next stage of our research. And like I said, we're interested in the CD90 positive and CD90 negative populations. I mean, the short answer is it's a work in progress. One of the important questions is whether we can kind of preserve the individual populations for long periods in vitro by whatever means, different scaffolds, different conditions, and how plastic these populations are. So we're beginning to do that, but we haven't got the data yet to discuss.
Terrific. And you have a question here from Hiro Ueno. I think you may have already answered this, but before he asked it, but what percentage of live cells can you get from cryopreserved samples?
So I reckon we lose about 30% as a sort of high end. So anything between about 15 and 30% once we've frozen and then thawed them out.
Okay. And question from Makey Jungin, where would you say the real added value of the ADTs is?
Well, I think so. I think in other areas of biology where you already have a lot of this information, it's probably not that helpful. But here, it's for me that the real use is in trying to see if we can have one or at least a collection of surface markers by which we can, as someone mentioned before, sort and isolate some of these cells and then interrogate them in vitro. And it's really difficult to know where to start with this. You know, those antibodies to surface markers that I put up were really the ones that BioLegend had available, and I mean, no one else really had anything to match those. But it's a repertoire we need to expand, and a lot of it's going to be trial and error and really just screening. You could say, well, why not use FACS? But you can, but it's difficult. FACS is difficult with these cells, and like I say, they autofluoresce to a high level, and they're so heterogeneous in shape that it can be hard to decide what is a positive and negative cell, even using isotypes. So I think, you know, CITE-Seq, the real advantage is you get a lot of information for the same experiment. You're not limited by autofluorescence, and it's a good way of screening lots of different surface markers. I mean, you know, FACS, you're limited to 14 at a time. There's no real limit to the number of antibodies recognizing surface proteins that you can use in CITE-Seq. You can go up to 30, 40. It's really only a cost that limits you.
Fantastic. Good to hear BioLegend again doing something that nobody else is doing. I love it. Let's see. We have maybe 5 more questions still coming in, and we'll cut this off before we get to the top of the hour here, so you can get on with your day. This question came in from Christian Gabel. Are the different tenocyte subtypes, are they present regardless of the source of tendon tissue?
Yeah, we do see quite a little bit of anatomic variation, and we certainly see a variation between individuals, certainly in the makeup and the percentages of each of those tenocyte groups. But in all cases, we saw representatives of each of the 5 clusters in all of our samples. So again, I don't think this is a good technique to comment on or to give an answer on proportionality or makeup. But we see examples in all our tendons. When we look at diseased and healthy, we see a predominance of the PTX3 positive cells in diseased tissue compared to healthy tendon. Although again, even in the healthy tendon, you can see examples of those cells. They're just very few. And it may well be that actually tenocyte A and B are probably a similar biological tenocyte, but A tends to dominate in disease, and it may just be that that's a cell that's changed or is driving or is part of the pathology of tendinopathy.
Okay, fantastic. We had a question from Kevin saying, very interesting talk, thank you. Actually 2 or 3 questions here in the same one. So saying, so chronically injured tendons, there are more M1 pro-inflammatory macrophages rather than M2 restorative ones. Is that true? And then also, what about macrophage function? Did you do pathway analysis, and where would were wound healing pathways unregulated in the MFs? Kind of a group of questions there.
I mean, I must say I'm not a macrophage biologist, but I appreciate there's some controversy in terms of using the terms M1 and M2. In our publication, we use those because one of the reviewers seem to be keen on that. It does seem that the macrophages dominate the immune cells that we could find ex vivo. But bear in mind that we do lose immune cells in our samples when we freeze and thaw the cell sample. So again, there may be other immune cells that we're missing. You're right, they do seem to dominate in the disease tendon, but again, it's not in terms of numbers or in proportion. It's more that they seem more activated, and we see a differential expression of genes that we'd associate with activated macrophages.
Great, and I think we'll go with 2 or 3 more questions here. Thank you so much for your patience and handling all these. We really appreciate it. This came in from Ruth, and I'll just go with the first name. Thanks for the interesting talk. Did you get any background sequence data from antibodies that bind several different cell populations that have low expression of the target epitope, such as lymphocytes, which have low expression of CD31? And if you do have background, how can you reduce it?
So we do see that. We do see a low expression level. It's not really an expression level, but it's a signal. We do see that. So for example, the ITGA7 positive pericytes have low expression background level of CD31 and CD34, which again kind of fits with their this idea that they may be arising from around perivascular structures. In terms of removing that background, it's not. I don't think it's as crucial as when you're, for example, using flow cytometry data and signals. It's normally much more clear cut. And again, you're dealing with logs of 2 powers. So although we do see it and it's interesting, at this very basic level of trying to identify surface markers by which we can begin to characterize our cells, it's normally pretty clear. Those that are expression levels, which is the term used, but it's normally a signal level of 2 or 3 times. That's normally enough for us to think that's a positive cell.
Okay, and then I think for the last question I see a couple of questions coming on the same subject here, so I think they're related. So let's go ahead with this. Someone's asking, was the amount of antibodies, or what is the amount of antibodies added for the CITE-Seq in comparison with flow cytometry? And someone else was asking, did you overload your 10x system with a higher cell concentration than the 700 to 1200 recommended? They noticed that the doublet seemed a little bit high.
Sorry, so the first part was the concentrations are pretty similar actually. Again, I'm sure that with time you could probably dilute these down to 1 in 50, 1 in 100, just like probably everyone did at the beginning of flow cytometry. But to start with, we basically used it as a sort of standard off-the-shelf concentration, and I think part of that is because the cost of sequencing is so high that it made sense really to not risk kind of losing that information. So but I think what's nice about the system is if you have any antibody that has a good pedigree or that you have good evidence binds well to your surface target of interest in your cell type, and again you can use in other cells that don't autofluoresce, you can use flow cytometry as a way of checking that, then there's no reason you can't use the same concentration of oligonucleotide conjugated antibody in CITE-Seq. It's not the fact that it's being sequenced doesn't make any difference to its binding properties. And what was the second part?
Second part was asking if you overloaded your 10x system. I'm sorry, I didn't realize that they were not related really at all. Did you overload your 10x system with a higher cell concentration than the recommended?
No, certainly not. I mean, we have very few cells compared to compared to other areas of where people are doing single cell sequencing. So no, in fact, we could run more. There's no reason why we couldn't put more through a single lane. So no, I think the doublets are interesting, and I wonder if I can't explain that. The other thing that we noticed is that there are quite a few cells that have bound hashing antibody, more than one bound hashing antibody. So presumably once we've hashed the samples and mixed them together to be run, I assume there must be some dissociation and rebinding of some of the antibody. And again, perhaps that's because the concentrations we're using were unnecessarily high, and maybe in the future we can drop that down.
Fantastic. I think we're going to go ahead and cut it off there. We're 5 minutes till the top of the hour. There were definitely some questions that we did not get to, so I'll forward those along, and hopefully we'll be able to respond in writing to as many of those as possible. I want to thank so much Dr. Kendal for presenting your work today. It was obviously, there's a lot of interest. This is probably the most questions I've seen on any of the webinars that we have hosted. So fantastic presentation. I want to thank BioLegend for sponsoring the webinar and for making it possible to share the content. I also want to invite everyone to our next webinar in this series. It'll be May 26th, and it will involve apoptosis and detecting and imaging apoptotic cells. So if you're interested in that, please join us. There will be a link to sign up for that in the follow-up email that you will get. And lastly, I want to thank all of you for attending. We still have about 100 people online after the entire presentation and question and answer. So thank you so much for being with us, and hope the presentation has been enlightening for any related work you may be doing in your own lab. If you do have any questions about the webinar content or regarding BioLegend or FluoroFinder products and services, please email us at support@fluorofinder.com. That's support@fluorofinder.com. And we will do our best to get back to you in a timely manner. Thanks again for attending, and we hope to see all of you online again very soon. Have a wonderful day.
Thank you. Bye.